October 1990, a worldwide initiative known as the Human Genome Project started to decipher the base pairs that are made up of human DNA in the task of identifying, charting, and sequencing all the genes within the human genome, considering both their physical and functional aspects. The Human Genome Project was originally led by James Watson and later led by Francis Collins. This monumental project was funded by the Department of Energy and the National Institutions of Health. Since this project was so big, it received help from 20 separate universities and research centers across the globe, including the United Kingdom, France, Germany, Japan, and China, and became known as the International Human Genome Sequencing Consortium. The major causes of the Human Genome Project was that learning and knowing the human genome sequence would facilitate an understanding of cancer, thus allowing a cure to be made, the desire to create different gene therapies, the idea of creating advancements in genome technology like automated genome sequencing devices, and lastly, trying to figure out the secret behind human evolution. This project escalated into a worldwide project with coming to the realization that to complete this project, one would need expertise from various fields and also have tens of thousands of workers. There was also a desire to create advancements in medicine to save millions of lives. The project later ended in April 2003. The Human Genome Project has contributed to the understanding and management of genetic diseases in various ways, as stated by Dr. Green.
What's happened over the last 20 years have been a series of things that have made the ability to sequence people's genomes more routine. And by studying the 3 billion letters of the human genome, we know more and more about how it works, although we have a long way to go. And getting to your question, we are learning more and more about how spelling differences in people's DNA, where you have one letter, I have a different letter, how that might influence our health, in some cases overtly causing a disease, in other cases just giving a greater risk for disease. And all of these things this past 20 years have been built on the basic accomplishment of the Human Genome Project, which is getting that first sequence of a human genome.
The Human Genome Project has made many contributions in this aspect, and these very contributions have led to cures being made for various diseases, such as a situation of two twins with a mutant gene. The Human Genome Sequencing Center at the Baylor College of Medicine found that there is a rare mutation in two twins that explain their mysterious, but increasingly life-threatening neuromuscular symptoms. Later, through whole genome sequencing, which the Human Genome Project laid the path for, the group at the Sequencing Center identified 3 genetic variants between the twins, which was further narrowed down to the cause of a mutation in a single gene, sapiopterin reductase, which would be the first step to finding a cure. Later, the Human Genome Project led to another discovery in 3 families that suffered from severe symptomatic calcification, which narrowed their leg arteries so severely that walking a few blocks would cause extreme anguish. Through genomic analysis, which the Human Genome Project laid the foundation for, it was found that there is a single mutation in the gene NT5E involved in calcium metabolism. By pinpointing the genetic cause, there has been several therapeutic approaches to the family's condition that NIH physicians are now developing. Another instance of the Human Genome Project playing a role in humankind understand and manage genetic diseases was its various contributions to cancer. More specifically, when US researchers were able to find a MSH2 gene which is linked to an increased risk of colon cancer, as well as researchers from Canada also finding variations on the FAD gene which is linked to Alzheimer's disease. The Human Genome Project, or HGP, proves highly valuable in advancing the field of personalized medicine due to most drugs only working on 30% of the population, and also there being differences between different genomes. The Human Genome Project helped to identify different genomes, which in turn allowed for more efficient drugs to be prescribed. Craig Venter, someone who played a major role in the Human Genome Project, used his genome to identify that he was at risk for heart disease, so he started taking statins to lower cholesterol, reducing his risk for heart disease. There is also a new application of technology now, including transcriptomics, which is RNA-based testing, metabolomics, which is analyzing the metabolites present in an individual, and proteomics, which is analyzing the entire complement of expressed proteins. Thanks to the Human Genome Project, we can use these technologies to create and identify genetic tendencies for diseases, which in turn gave us the ability to make personalized treatments possible. In another study that analyzed the DNA of 22 patients with lung cancer to reveal whether the patients had extra copies of the epidermal growth factor receptor, the EGFR gene, it is known that patients with this gene respond to certain types of drugs, including gefetinib and erlotinib, which means that with this information, we can develop more specialized and successful treatments. By figuring out our genetic makeup, the Human Genome Project made great advancements in gene therapy. The investigation uncovered genes linked to many illnesses, which laid the groundwork for specialized gene therapies. In September 1990, William Anderson headed a team that carried out the first gene therapy clinical trial, approved for the use in a human. The goal of this phase 1 study was to define the safety issues involved. The patient was a 4-year-old girl who had a genetic disease called ADA deficiency, which is caused by a defective gene for the enzyme ADA, resulting in SCID via a modified retrovirus. Normal ADA genes were transferred to T lymphocytes that had been removed from the girl's body and grown in culture. The white cells then were returned to the patient. The Human Genome Project has also identified genes for various other diseases that have or could eventually lead to a cure, stated by Dr. Eric Green.
What are known as rare diseases, and at the other end is common diseases. So, because progress has really been substantial with rare diseases, and we're going to see more progress in the next 10 years with common diseases. Now, what's the difference? Rare diseases are diseases like sickle cell anemia, cystic fibrosis, Huntington's disease, and so forth, that are caused by mutations, DNA changes in a single gene. You break a gene, you get the disease. When I say we've made progress, I'll give you a real number. The day the Human Genome Project began, there were 61 rare diseases that we knew what the gene that was mutated in that disease. 61. One of them was sickle cell anemia. So before the Genome Project began, 61. Today, it's about 6,000.
The Human Genome Project also found a few genes for breast cancer, which could lead to cures in the future. The Human Genome Project has provoked a range of viewpoints, including those pertaining to ethics, society, and culture. While proponents of ethics and privacy emphasize protections against genetic discrimination, indigenous communities emphasize the significance of collecting genetic samples in a way that respects their culture. Advocates for social justice draw attention to the necessity of equal access to genetic advances and benefits. Religious organizations voice moral doubts, while anti-technological movements add to the skepticism. Acknowledging this diversity is essential, and continuous efforts are concentrated on policies and ethical guidelines to address issues and guarantee responsible genomic practices. A comprehensive approach to these ethical, legal, and social dimensions of the Human Genome Project requires the integration of these perspectives. And though the Human Genome Project has been questioned by many different groups, its results are undeniable and it has saved many lives. This project has affected us in a way we've never seen before. The Human Genome Project marked a seismic shift in our understanding of the human genome. From 1990 to 2003, it meticulously mapped and decoded many human genes. This monumental endeavor laid the groundwork for personalized medicine, offering insights into individual genetic profiles. The Human Genome Project reshaped our understanding of genetics, mapping every human gene. Ethical discussions on privacy emerged, fueling bioethics. Scientifically, the Human Genome Project's data advanced genomics, influencing health approaches. The Human Genome Project stands as a transformative turning point in genetics and genomics.